DNA. We talk about it so much because it's the ultimate director for cells and it codes for all of your traits. With a molecule that has a function like that, it makes sense that when you want to make another cell, like you do in cell division, that you'd also want to get to need more DNA into the new daughter cell. And that introduces our topic for today, DNA replication, which means making more DNA. But first, let's talk about the where and the when DNA replication occurs. Where? Well, if it's in a eukaryotic cell, it's going to occur inside the nucleus. But remember, not all cells have a nucleus, such as the prokaryotic cell. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells, they do have to do DNA replication, but there are some differences that we're really not going to get into today during this demo lesson. Next, the when. When does DNA replication happen? So if a cell's going to need to do this, it's going to happen before the cell divides so that the new daughter cell can also get a copy of the DNA. In eukaryotic cells, that's going to be before mitosis or meiosis in a time called interphase, which is when most of the cell's life cycle is spent in interphase. You know, I kind of think DNA replication would make a pretty cool video game, but we're all kind of still waiting on that to be invented. Let me introduce you to a few key players in DNA replication so that we have some background going into things. Now remember, these are just a few key players because it's a super complicated process and would take way more than 30 minutes. Many of the key players are called enzymes. Remember in bio, if you see something that ends in ASE, probably it's likely going to be an enzyme. Remember, enzymes have the ability to speed up reactions and build up or break things down, such as the items that they act on in the reaction. So here we go with our key players. We have helicase. Helicase is the unzipping enzyme. If you recall that DNA has two strands and you can think of helicase as unzipping the two strands like a zipper of DNA, it doesn't really have a hard time doing it as it just kind of breaks through the hydrogen bonds that hold those DNA bases together, the A, the T, the G, the C. So there's DNA polymerase, which is the builder. This enzyme replicates DNA molecules and actually makes a brand new strand of DNA. Then there's primase, the initializer. With as great as DNA polymerase, it can't really figure out where to get started without something called a primer in the code. So primerase is the things that makes the primer so that DNA polymerase can figure out where to get started on its work. You know what's also kind of cool about a DNA primer? It's made out of RNA, which I'm assuming you've talked about in previous lessons. Then there's ligase, the gluer. Ligase helps DNA glue back together. We'll talk a little bit more about it in a little bit, but I don't want you to feel overwhelmed because we're going to go through the basic sequence of it in order, okay? So, but the important thing to take away is that this is just an overview. This is in no way going in depth about what really goes on. And just as a reminder, ligase doesn't actually go around with bottles of glue. So, let's get started. So DNA replication starts at a certain point called the origin. Usually this part is identified by certain DNA sequences or a code line. At the origin, helicase, which is that unzipping enzyme, remember, it comes in and it kind of unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So we have these things called SSB proteins that are single-stranded binding proteins that bind to the new DNA strands and it keeps them separated. Then there's topoisomerase. You got to kind of slow down to say that, topoisomerase. Topoisomerase, it keeps the DNA from doing the supercoiling, which it might sound kind of cool, and it kind of is, but if you're trying to compact DNA, it's something that has to be controlled, or else you can kind of overwind the DNA. And remember, you need the DNA strands to sort of be separate for the next steps to occur, which is transcription and translation. So primase comes in and makes the primers, and so it knows where to get started. Otherwise, DNA polymerase does not know where to begin. So then in comes our DNA polymerase after our primer is laid. But I want to go into one thing. Before I move on, remember how DNA has two strands? they're not identical, but they complement each other. Well, you want to think about how are they connected? How do these base pairs pair together with hydrogen bonds? Remember, adenine goes with thymine, A with T, and guanine goes with cytosine, G with C. But these strands are also antiparallel, meaning that they don't go in the same direction. So what do I mean by different direction? But when we talk about DNA, we don't really say like north or south. We say DNA either goes from 5' to 3' or 3' to 5' direction. But what does that mean? Well, the sugar of DNA is part of the backbone of DNA, like the spine. It has carbons on it. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and then 5'. The 5' carbon is usually outside of this ring structure by this drawing. Now you do the same thing on the other side, but keep in mind that the DNA strands are anti-parallel. So let's count these. Clockwise again, starting after the oxygen, 1', 2', 3', 4', and 5' carbon. And the 5' is out of the ring again. This strand on the left here runs 5' to 3', and the strand here on the right runs from 3' to 5'. I'll explain why that matters in just a moment. So let's take a look at the knowledge and look at DNA replication. In this image, notice that the labeling of the top original strand is 3' to 5'. The bottom original strand is labeled 5' to 3'. That's the original DNA that is going to be replicated. DNA is winding here thanks to our enzyme friend helicase. In this example, it will keep unwinding in this direction. But primase places primers. DNA polymerase is building the new strands. Now, there's thing about DNA polymerase is when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning that it adds new bases to the 3' end of the new strand, that is. So see how it's being built from the 5' to the 3' direction? This is known as the leading strand. But take a look down here. DNA polymerase, once again, is building a new strand in the 5' to 3' direction. But there's kind of a problem. Let's take a look. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5', 3' direction, it has to keep kind of racing up here to where it's unwinding, where that's happening. You can see why this is the new strand is known as the lagging strand. On this lagging strand, the primers have to keep being placed in the order for the polymerase, DNA polymerase, to kind of keep building. And these little fragments that are made are called Okazaki fragments. I don't know, kind of a funny name. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I kind of nicknamed it, has to take care of those gaps between those Okazaki fragments, which seals them together. At the end of this replicating, you have two identical double helix DNA molecules from that one original double helix DNA molecule. We call this semi-conservative conservation because two copies each contain one old and one original new strand.